the mechanism which is involved here is, one is the DNA methylation, any change in the histone, that is like histone modification, and rcRNA, that is non-coding RNA. Just see what is this methylation. This methylation is the one common things which is happening nowadays in our day-to-day life because of the stress and all this thing, and that can lead into different imbalance in the lifestyle. So the DNA methylation is one of the most probably studied things in the nowadays what we are facing, what we are going through. Back in 1969, Griffith and Miller, these are the two people who have found that yes, DNA modification or any changes in DNA is happening is mainly due to the DNA methylation. So now it's a common type of epigenetic modification. What we call it as a methylation is very common nowadays. This methylation involves attachment of a small molecule, a small or chemical group, which has a, you can see here, a carbon group and a 3 hydrogen group in it. Okay? So this a small compound will go and bind to the DNA. I don't know if you can able to see my cursor. See, if this is the DNA double helical strand. You can see 2 small blue dot. So these called the epigenetic capping. So now this particular sequence is marked. The second epigenetic modification is by histone modification. Histone modification are proposed to affect chromosome function through at least 2 distinct mechanisms. So the first mechanism suggests that modification may be altered due to electrostatic charges. See, the histones are having 2 different charges. So with the positive charges, which will be on the core of the DNA, and the tail of the histone will have a negatively charged. So any electrostatic disturbance here, so this entire chain is disturbed, and then comes the issues in the DNA. The second mechanism is the way they bind. So histone proteins are so important to make sure that the large DNA molecules are supercoiled in such a way that it are supercoiled, and this supercoiled thing are packed inside a cell. You know, when you stretch a DNA, it can run to a mile. So this much of DNA cannot usually it will be present in a small cell. That is because the supercoiling of DNA. So the supercoiling of DNA is done by the histone protein. So histone proteins are so important to make sure that such a big strand are compiled or they are archived in such a way that this information is sit inside a small cell. So histone is very important. At the same time, the DNA strand is very important, where you have a different genes in it, which can result in different proteins or different representation of production of proteins. Okay, now what happens, let us see, when there is no histone, DNA would be too longer to fit inside a cell. That's what we discussed. If histones are squeezed so tightly, they cannot be read. So modification that relax the histone can make the DNA accessible protein and then read. So now the switch off and on mechanism will happen. Now if you see on the left hand side, so here you can see there is a supercoiling of DNA where the histone proteins are jam-packed. So methylation of DNA and the histone causes nucleosome to pack tightly together. So you can see this here. The nucleosomes are so tightly packed together. So that because of this particular strand of DNA is inactive. We cannot read it. So the necessary factor, the positive and the feedback mechanism which a system should follow, this becomes will be mainly controlled by this thing. So when the genes are inactive, so there is likely cannot be read. But here on the other side, histone acetylation result in loose pack of nucleosome. So these genes are expressed. So what all this particular gene is meant for, that is read, and accordingly that what all the derivatives of that particular gene is active, and that will be produced in our system. So this is how the positive and the negative feedback mechanism will happen in our system. Now, if you see this thing, there are some epigenetic factor. The epigenetic factor could be an inheritant or induced by the environmental changes. So this will affect on the DNA. So on this DNA, you have a small capping, which called a DNA methylation. So DNA methyl group, it's an epigenetic factor, which will go and anchor in that particular sequence of DNA. Now, if you see here, the positive and the feedback mechanism, just what we were discussed about, histone proteins are histones are protein which are wrapped around the DNA, and when they are tightly packed, the particular gene is not read, so that there is a negative feedback. So when a particular gene or anything has to be produced, it will unwind itself, and then comes the positive feedback mechanism. So if you see all this thing, there are an epigenetic factor always anchoring on the particular sequence of this gene. Either it could be on the strand of the DNA, or it could be on the histone protein. Okay. Now coming to one of the third epigenetic factor, which is called microRNA or a non-coding RNA. Does ncRNA represent a small RNA molecule encoded in the genome of the plants or the animal? This highly conserved 22 nucleotide long RNA sequence regulates the expression of genes by binding into the 3'. As we know, the DNA has a double helix. One runs from the 5' to 3'. The other is from the 3' to 5'. So now, okay, this I will show here. So here is the double helix DNA. So now when they go for a transcription, the DNA unwinds, the messenger RNA comes, and what are the necessary proteins which are meant to happen will be produced. And this protein is used for the homeostasis of the system. Now this microRNA will go and bind 3' of it, and it stops the production of protein. So that's how the positive and negative feedback will happen. So any proteins or any molecules in our system should have a restriction. Like when it has to produce, it should produce only that period. When it has to cut off, it should cut off. So this is how the positive and the negative feedback mechanism will happen.